Hi, Assalamualaikum. My name is Siti Murzaifa binti Mursalin. My matrix number is S52103. Today I want to present about the polymerase chain reaction for AQU 3601. Okay, first I want to tell about the history of PCR. This PCR is invention from Carey Mullis at 1987. Okay, he is received the Nobel Prize in Chemistry for discovery of polymerase chain reaction, and he also breakthrough in expanding the limits of knowledge in molecular biology. The next is I want to tell what is PCR. As you know, PCR is a technique used to make several copies of a small fragment of DNA and mRNA. Okay, next is PCR is in vitro technique for amplification of DNA, molecular biochemical technology. DNA strands are used as a template, size and sequence of fragment defined by primer. Okay, PCR words made up of two words is polymerase and chain reaction. For polymerase reaction is enzyme. Enzyme is the one makes a new strand such as DNA polymerase. For the chemical reaction, process in an exponential way and copying million of DNA. Okay, this is for the first reaction. Okay, this is a chemical reaction. For first reaction, can make 2 molecules of DNA. For the second reaction, make 4 copies of DNA. For the third reaction, make 16 copies. And the next step will make 33 copies. And the next reaction is 64 copies, and so on. Reaction will make million copies of DNA or billion copies. Next, this is application of PCR. First is fish disease diagnostic, DNA cloning, population genetic, phylogeny analysis, and DNA sequencing. Okay, they have 4 functions of PCR that I have understand. First is to make multiple of copies of a segment of DNA to amplify a specific DNA target for mix of DNA molecules for wide variety of applications such as genotyping. The last one is to detect of a carrier fish in broodstock from a farm. Okay, they have 2 factors of PCR. First is magnesium. Magnesium is for the DNA polymerase to function. Okay, they have 2 concentrations in the magnesium that can affect the PCR. First is the low concentration, where when the low concentration of magnesium, the polymerase is insufficiently active. But for the high concentration, the PCR loses specificity and multiple products are produced. Okay, the second one is oligonucleotide primer. Yeah, this is PCR experiment was wholly depend on the oligonucleotides primer. So the individual primer should now contain sequence that are complementary. Okay, they have this is I want to tell about the process of PCR that very important to make our DNA successful for the new strand. And first is this I want to tell about the component in PCR. That very important is first is chemical reagent. Chemical reagent they have 1, 2, 3, 4, 5, 6, 7, 8 chemical reagent that must be in the PCR to make our DNA is successful. Okay, next is total volume of PCR mixture thin wall tube, and the PCR machine is a thermocycler. Thermocycler, they have a DNA sample, primer, nucleotides, PCR tube, mixed buffer, and tag polymerase. In the thermocycler, tag polymerase is a type of DNA polymerase and like DNA replication. In PCR in PCR. The type polymerase also called as a dark polymerase. Okay, for the DNA, okay, DNA template is original DNA that we must to amplify. Okay, for the nucleotides is the basic building blocks for DNA synthesis. Okay, next is this is they have X step for PCR cycle that we must to know. Okay. Okay, there's 3 steps in PCR. First is denaturation, annealing, and extension. For the denaturation, we must using 94 Celsius until 96 Celsius for 2 minutes. Okay, the denaturation function is to denumerate or separate the 2 strands of DNA. And then the result we get 2 strands of DNA. Okay, such as DNA can be reversely denatured by a cycle of heating and cooling. Okay. Next is annealing. Annealing is this word we also hear from the industry, from the metal industry. This function is to remove the internal defect. Okay, for the annealing, we must use 72 Celsius or range 55 until 60 Celsius will be chosen for this process. Okay. Okay, for the cool temperature, we must use for 55 Celsius. So the primer in the PCR tube can be bind to the target sequence on the single strand of DNA. Okay, the primer sequence matched to the starting points. So in the result of the annealing, we can get 4 single strands of DNA. Okay, for the extension is we must increase the temperature of at 72 Celsius for the PCR process. Okay, in this process, the polymerase extends the primer synthesizing new strands of DNA, and then the polymerase adding new nucleotides to the short sequence of the primer to form new strands of DNA, and then this tube along with the cofactor needed by enzyme and repeat cycle of heating and cooling to allow DNA. So we can get add new strand of DNA in this extension process. So also we can use this 94 Celsius for the initiating step and still retain full activity. Okay. Okay, the enzyme does not need to replenish during the PCR reaction and heating cooling cycle set. Like I said, because for PCR to proceed can be fully automated in specially designed heating block. And the high temperature at which extension reaction can be performed means that specificity of primer annealing is not compromised. This is product of PCR. So this is DNA primer. This is a PCR product. We can know the kilobase pair of DNA, and then this is a forward primer and reverse primer. Okay, they have 2 types of PCR. First is reverse transcription. Okay, this is method for RNA amplification and quantization after its conversion to DNA. For RT-PCR, consists 2 parts. First is synthesis of DNA from the RNA by reverse transcription at 42 Celsius. And second one is amplification of a specific DNA molecule by PCR. Okay, next is real-time PCR. Real-time PCR is quantitative reaction products for each sample in every cycle based on Ct value. Like, okay, real-time PCR system rely upon the detection and quantitation of fluorescent reporter. That's all for me. Thank you.